Thank you very much to Professor Yuval Neria. We will save the questions for the end of the panel so we can have also some kind of discussion addressing all the three talks that we're going to hear. So I am delighted to invite our second speaker, Professor Ruth Feldman. Professor Feldman is a Sims-Mans Professor of Developmental, Social Neuroscience, and Director of the Center for Developmental, Social, and Relationship Neuroscience at the Interdisciplinary Center at Herzliya, with a joint appointment at Yale University Child Study Center. She is also Director of the Irvin B. Harris Community-Based Clinic and Internship Program for Young Children and Their Families. Her research focuses on the biological basis of social affiliation, processes of biobehavioral synchrony, longitudinal follow-up of infants in high risk stemming from biological, maternal, and contextual risk conditions, the neuroscience of empathy, and the effects of touch-based interventions. Her studies on the role of oxytocin in health and psychopathology have been instrumental for understanding the biological basis of social collaborations in humans. Furthermore, Professor Feldman's research on the maternal and paternal brain-human bond formation, the long-term effects of kangaroo care on premature infants, the brain basis of conflict resolution, and the effects of maternal postpartum depression on children's brains and behavior received substantial empirical and media attention. Professor Feldman is a fellow of the Association of Psychological Science and has been on the editorial board of several high-impact journals. She has published over 300 articles in scientific journals and book chapters. It is with great honor to invite Professor Feldman to present her talk about the effects of chronic war-related trauma on the developing brain and infant-to-adolescent study. Professor Feldman.
Good morning. It's a real honor to be here and speak in honor of Zahava and her immense impact on the conceptual models and empirical finding on trauma. Well, I think trauma has been in the inception of mental health since the beginning. If you remember, Freud's first model was affect trauma, when he thought that these hysterical women that he saw in Vienna really suffered trauma. And actually, psychoanalysis is the shift from real trauma to mental trauma. But I'd like to follow Professor Neria's talk and say that when the neurobiology of research of trauma has started to be explored, we focused on fear circuitry, the amygdala, prefrontal cortex, whatever you heard before, conditioned stimulus. This is understandable. Fear lies at the heart of the traumatic experience, but it could also be very easily modeled in animal research. Humans have not evolved very much from rodents when it comes to our immediate fear response. And what I'd like to offer you today is a different window. I'd like to present an infancy-to-adolescence study of children in Sderot that focuses on a different framework to PTSD through the oxytocin system, through behavioral synchrony, and through the neural basis of empathy. And here we shift the focus to include another system that, as I'll argue and show you today, is no less ancient in terms of its evolutionary origin than the fear response, the parent-infant bond as the social buffering mechanism, and what it does to the development of the social brain, tilting a little bit of our, not only broadening our neurobiological underpinnings of trauma, but also focusing a little more on resilience. What differentiates children or adolescents exposed to trauma who develop PTSD or other psychiatric disorders from those who are exposed to the same exact conditions who are more resilient? And we're looking for early precursors of resilience. So it's focusing more on the resilient and the relational and the empathic biological, which is no less biological underpinning, but expanding the scope of search to include the affiliative response, not only the fear response. So just two images to open. This is the famous monk picture, a small figure exposed to horror unspeakable horror preceded, maybe foreshadowed, the bloody 20th century. And here is exact same expression from a little kid, Shelley, 3.5-year-old, one of our subjects in Sderot. And the question is how these kind of fear and horror a child faces, what modulated it biologically, brain-wise, and how it could be contained. This is our model. I will tell you about a 15-year study of children in Sderot, Israel. And let me see, where's the pointer? Okay, so I don't. Okay, got it. As you could see, Sderot is 10 kilometers from the Gaza border. Individuals in Sderot have been exposed for the past 20 years to repeated missile attacks that come chronically, unpredictably. Every couple of months there is an exacerbation of the situation. So we have military operation. During military operation, there are hundreds of Scuds, missiles, and recently fire balloons. So the experience is continuous war-related stress, unpredictable, chronic. The children we follow have been born to this situation, so it's prenatal stress. But moreover, in terms of human research, our study offers a natural experiment that is very rare in human studies. Because when you look at early life stress or other trauma in children, such as domestic violence or poverty or other sorts of abuse, one child, one family is very, very different from the other. Here we have a very unique case of war-related trauma where all the children are exposed to exactly the same condition, same trauma. And we ask what differentiates Uri, who lives in Hakalanit 2, from Dani, who lives in Hakalanit 4, and what are the factors, either in their biology or their temperament or their genes or the parenting or anything familial that differentiate those exposed to exact same stressor who are on a more risky trajectory or are more resilient. And this is a very, very unique follow-up enabled in Sderot. This is our study. We have done 5 time points in early childhood, middle childhood 6 to 8, late childhood 9-10, early adolescence 11 to 13, and late adolescent 15 to 16. And we're now preparing for the study at emerging adulthood. And these are the measures we took, and whatever is underlined and bolded is data from this time points and measures that I'll show you today. So I'll show you diagnosis measures, interaction measures, hormones, genes, and imaging by MEG at the transition to adolescence. So just to speak about our system, the oxytocin system. The oxytocin system is a very ancient system. It's presumably evolved from gene duplication in jawfish approximately 600 million years ago, and its goal was to enable organisms to manage in harsh ecologies. And it's implicated in ancient organisms, from nematodes, C. elegans, early organism, in basic regulatory functions such as energy conservation, water balance, or thermoregulation. With the evolution of mammals, oxytocin became implicated in birth and lactation, the 2 mammalian functions. It's implicated in uterine contraction during birth and in milk lectine during lactation. But with it, the main mechanism for stress buffering became the mother-infant bond. So mammals manage stress through the relationship with the mother due to the ancient oxytocin system and its evolution and incorporation into the major mammalian functions. So here we speak about a 9-amino acid neuropeptide oxytocin with integrative functions. It's closely linked with the HPA, the stress response, with the dopamine, with the immune systems. Receptors are widely distributed throughout body and brain, so it's an integrative system. It has strong epigenetics effect. It's shaped by early social experience, and parent-infant synchrony and parental OT longitudinally shape the child's OT. So you get both from the genes of your parents, but also very, very strongly through experiences of biobehavioral synchrony. And what is biobehavioral synchrony? It is defined as the coordination of biology and behavior among affiliated members during moments of social contact. So we found in mother-infant interaction that during moments of their interaction, when mothers coordinate with their infants their behavior in the gaze, affect, vocal, and touch modality, there is also a coordination of heart rhythms. You find it in when they behaviorally synchronize, they also coordinate their heart rhythms, and it's not found in non-synchronous moments. There's a coordinated release between parent and infant in their oxytocin level, and there is brain-to-brain synchrony during moments of behavioral synchrony in key nodes of the social brain. So behavioral synchrony is a behavior-based, bottom-up mechanism where behaviors, species-typical behaviors within the bonding context, the maternal behavior, and then the mother-infant coordination, they shape the organization. They provide external regulation to these environment-dependent systems that support stress by affiliation, the autonomic system, the oxytocin system, and the maturation of the social brain. The experience of synchrony is critical during the sensitive period, and it goes both lengthwise and widthwise. Lengthwise, we found that during the sensitive period, the experience of synchrony between birth and 9 months, then later, like ripple effect, is predictive of the same synchrony between mother and child as they grow older. This is a study from birth to 22 years, and you see that as new abilities is incorporated into the mother-child relationship, more symbolic, more conceptual, more empathic ability, the ability to dialogue multiple perspective, they are built upon and integrated into the early nonverbal synchrony of the sensitive periods before infants learn to talk. But it's also synchrony experienced within the mother-infant bond also builds the brain structure, the hormonal system, and the machinery for other synchronous human relationship, not only with the parent, but also with other. So if we speak about behavioral synchrony in the nonverbal, in the gaze, in the affect, in the vocalization, in touch patterns, we speak about heart rate synchrony, hormonal synchrony, and brain synchrony. We found evidence for all of these types of synchrony in all humans' relationship, between parents and infants, between romantic partners, between close friends, and among strangers, fellow humans. Humans are the only species where we can activate our affiliative biology and our empathic brain to the distress of strangers. This is not found in rodents or primates who need a period of familiarization in order to be empathic to the distress of conspecific. Now, what is important is, in the context of our Sderot study of the PTSD, we look for synchrony not only during positive social interaction between parents, mothers, and infants or children or adolescents, but also how mothers synchronize and provide external regulation of stress. So in early childhood, we ask mothers to tell us about the trauma when the child is present. So we interview the trauma. It's like trauma reminders when the child is present, and we see not only the child response to trauma evocation, but also the mother's synchronous behavior towards the child. In middle childhood, by the way, we had a parallel study in Nablus and Jenin to the PTSD study in Sderot, but it was stopped about a decade ago. We couldn't continue beyond age 5. In middle childhood, we asked them to co-construct a narrative through book reading of a stressful story. And in adolescence, we ask them to dialogue a conflict. So we don't look for synchrony only in positive moments, but also how mothers modulate stress and provide the stress regulatory finding. So let me show you some finding from our longitudinal, some among the many, related to the oxytocin system and its role in risk and resilience, and then the maturation of the empathic brain in children exposed to war-related trauma and in their mothers. So just to show you about the risk and resilience, we initially recruited 238 families, 148 from Sderot and 84 matched controls. In the early childhood, we had a third of the children who were diagnosed with PTSD according to the DC 0 to 5, so a full-blown PTSD in a third of the population. It's like quite a high number, much higher than we found. But we had 2/3 of the children who were exposed to the same stressor and were more resilient. And what you see is we saw 4 types of reaction during the time when the child is exposed to the mother narrating the trauma evocation. This happens to be the worst, the avoidant reaction. I think a lot of Zahava's work would attest to that. This is a child in a fetal position, going away from the mother and the experimenter to the door. Our feeling is that if he could go any farther, he would leave completely. These children do the worst. These children with hyper angry and anxious reaction, middle, and those do the best. So actually a regressive response, a child who shows proximity seeking, a child who clings to the mother during trauma evocation, these are the children who eventually would do better. And this is important to know, that children who don't make trouble in kindergarten and primary school are not those who are on a resilient trajectory. These are on the worst trajectory, as seen later by the cortisol stress response, the oxytocin response, their symptoms later on, and their brain. So we do need to pay attention to those children and to those children. And children who seek their mothers, usually it's a sign of resilience. This is the oxytocin receptor gene. It has 17 SNPs, and 4 of those SNPs, the 298, 778, 576, and the CD38, which is a molecule implicated also in immune processes and is responsible for the release of oxytocin to the pituitary, and the vasopressin, which is a sister molecule. And what we did here, we genotyped mothers, fathers, and children. Okay, genes is the only thing that fathers in Sderot in the sample were willing to give us. Despite repeated attempts, they weren't willing to engage in anything else, even though we tried, but mothers have been with us through the 6 times. So we measured each variant come in a more risky format and a more resilient format, which means that there is, in the risky format, there is less oxytocin in the brain. And we computed a cumulative risk score, which ranges from 0, that you have no risk, to 5, that you have all your SNPs are risky. So you can get between 0 and 5, anything for the mother, for the father, and for the child. And we want to see what is the role of growing up in an oxytocinergic or oxytocin-filled environment and the child psychopathology. What we saw at first, we're talking about middle childhood, so children here are between 6 and 8 years old, and this number is really staggering, because 60% of the children who were growing up in the context of war-related trauma developed, by the time they are 6, a full-blown Axis 1 psychiatric disorder. 60%. This is really huge, and I think Zahava's work would show us that this doesn't tend to go away. That even if, let's say, the numbers drop to 50% by the time there are, or maybe 48% by the time they are 16, those children are still at risk, even though they had a diagnosis at 6 years and no longer have it by 16. Moreover, these children, unlike our other cohorts, like growing up with a depressed mother or or being born prematurely, those children tended to have a lot of comorbidities. So more than half of those children had more than one disorder, like PTSD and an anxiety disorder, or PTSD and a conduct disorder. So this is high risk, and we're talking in relation to approximately 15% in the general population. And you see the main players here. We have a lot of children, like 28% of the children with PTSD, anxiety disorder, conduct disorder, and ADHD. So this is of note. But what are the factors related to risk and resilience? What does the oxytocin system has to do with it? So here we see that when children are at genetic risk, meaning that there is less oxytocin in their brain, there's more genes that spell risk in the child, those children who are with the risky oxytocin system tend to score much higher on psychiatric disorder than those, and those children who receive more maternal support during those traumatic situations. These were 30 minutes where the mother was evoking the trauma, and we saw how she behaves towards the child. Some mothers who gave more support than others, this is a behavioral coding of blind coders, their children tended to show much less psychopathology later on. And then we went to see which children had chronic PTSD, we're talking about early childhood to middle childhood, and who are the children that remitted. And here you see amazingly that the factors linked to the resilience, to being remitted, having PTSD as a young baby, and then recovering. It's the genetic risk score on the oxytocin pathway gene, on the extended oxytocin system. So you see here that mother's, child's cumulative risk, father cumulative risk, and mother's cumulative risk were all related to how much the child is prone to psychopathology. So here we have our first evidence showing that this is a system that's involved in differentiating children exposed to exact same stressor, to a trajectory that's more risk, and to a trajectory that's more resilient. And again, maternal support also differentiated children who were chronically PTSD from those who are more remitted. Okay, so maternal, paternal, and child genetic risk and maternal support differentiate chronic from remitted PTSD. And now I'd like to show you two studies, two MEG studies. This is Yoni Levy, Dr. Yoni Levy, our imager who ran all these studies. And what we did was, at the transition to adolescent, children are between 11 and 13 years, we imaged both the mother and the child, considering that since the maternal oxytocin system and the mother-child synchronous relationship is so critical, we want to see what happens in the brain of mothers who need to raise children in the context of chronic stress and unpredictable war. So I'll show you, I'll say a few words about the empathic brain, and then I'll show you one data set from the child and one from the mother, and you'll see the role of synchrony in both. So basically, empathy, studies on the neural basis of empathy, the brain basis of empathy, have used, Simon Shamaitsuri wrote about it, among others, talking about a two-pronged process. One that's more automatic, nonverbal, bottom-up, more the orange, what you see, what's called the mirror neuron system or the embodied simulation system, the system that responds to the motor movements of someone else, and a more mentalizing component, the blue parts, the superior temporal sulcus, the superior temporal gyrus, the VMPFC, posterior cingulate, precuneus, areas of the DMN. So here you see two systems, one more primitive in our gut response to someone's distress, and one more mentalizing, that's calling to action, understanding the other person's perspective, and also differentiating self from others. However, in a recent meta-analysis that Franz de Waal published in Nature Neuroscience last year and summarized all studies on empathy, he spoke about a certain widely distributed area, particularly the SMA-MCC, which is shared by the two. So more ecological studies that exposed individuals in the magnet to more ecological conditions showed activation of these two. And really you can understand it because when you are in real life responding to the distress of other, you don't differentiate your gut response from your brain response. You really do both at the same time. So here is our study. In early childhood, children are between 2 and 3. We did mother-child synchrony during free play, but we also looked for 2 types of temperamental reactivity. The name for them in developmental research is the orchids and the dandelions, children who tend to be very fearful and very reactive to their early environments, show a lot of negative emotionality to early stimuli. It's a temperamental, biologically based trait. From children who are dandelion and can grow anywhere and are less reactive. At T3, we again measured synchrony during a mother-child discussion paradigm, and we had a psychiatric evaluation of children's disorder. We focused on internalizing disorder because we know from past research that children with high temperamental reactivity tend to develop more anxiety disorder later on in childhood and adolescence, and we wanted to see what war and stress have to do with it. And at time 4 in pre-adolescent, we exposed children to an ecologically valid 5 minutes, I'll try to finish at least this, paradigm, and particularly looked for the shared component of the bottom up and top down, and the mirror of the mentalizing network. So you see here is the low negative, it's a very common fear paradigm, the mask paradigm, and the highly reactive child. This is the paradigm. We described empathy, situation that are typical of children. This child, his friends don't want to. We first asked the children to try to take the vicarious perspective, the perspective of the other, and then we had several situations that where this child, his friends don't want to talk to him, this child is afraid he's going to fail an important test, versus control battery, this child is reading a book, or this child is tying his shoes, etc. And let's see what happened. So let me walk you through it. The first thing we found that there is activation in the alpha range, and this is the brain's default oscillation. This is what to be expected. And here you see a widely distributed response in the empathic brain in those areas that are shared between emotional and cognitive empathy. We found no difference in the level of activation between the Sderot and the control, but we found difference mediated by synchrony. So we see it spread, it divides to risk and resilient. Children who received synchronous parenting had similar to others, and children who received less synchrony, generally there was less synchrony between mothers of the Sderot kids, had low. So there's no direct effect of war exposure on the empathic brain, but effect that is mediated by mother-child synchrony. What happened to the fearful children? When we looked only at activation in the exposed children, at the Sderot children, we saw additional areas that we initially thought are part of the mentalizing, the cognitive empathy. Those activations were only found in the fearful children in the Sderot. They were not found in the low reactive, in the dandelion of Sderot. Those children tended to develop internalizing disorder in late childhood, and internalizing disorder was associated with this activation. And we concluded that those activations don't have anything to do with empathy, but to do with the rumination and inability to disengage the brain basis of anxiety. Those children are unable to disengage from negative stimuli. I will go very quickly through what happens to the mother's brain. This is a empathy to pain paradigm. The adult brain, but not the child brain, shows activation, additional activation that develop only in adulthood, in gamma rhythm, in what we call the visceromotor gamba, in areas that are implicated in interoception, the ability to feel the internal milieu of the body and use this feeling for empathy with a situation of other. This only develop in adulthood. We had a large study of 209 children, adolescents, and adults, all healthy, and we found that the empathy moves from sensory to mentalization and from single to multiple rhythm. And what you basically see here, that this visceral motor gamma that enable mothers to do higher order cognitive empathy, use their experience of their own bodily milieu in the service of being empathic to other, is not found in the exposed mother. Meaning that their brain basis of amdan, brain basis of empathy, is just like a child or an adolescent. They haven't developed this higher order mechanism to be empathetic from far, from a cognitive perspective. Both gamma rhythm and the visceral motor cortex are signs of maturity. This is how it looks. And the mother's gamma rhythm is related to both mother-child synchrony, the experience of empathy, and to the child prosocial skill, and this is a full moderation model. So you see exposure leads to lower mother-infant synchrony, mother-child synchrony throughout the first decade. This leads to lower maternal gamma activity, a sign of maturity, and that leads to lower child prosocial abilities in adolescence. So here you see another way of not only the child's brain is impacted by mother-child's behavior, but also the mother's brain is impacted by mother-child interaction, mother-child synchrony, and that in turn impacts the child's prosocial ability. And to conclude, the child empathic brain is built through the experience of mother-child synchrony, which provide repeated practices in interactions that foster interpersonal resonance within the mother-child bond. The neural empathic response to others' emotional distress is a distinct process from mere emotional reactivity. This additional response in areas preventatively considered to be part of the mentalizing network may index over-mentalization, rumination, and inability to disengage from negative stimuli. And just 3 future directions. We need to move from the neurobiology of risk to the neurobiology of resilience. We need to develop models of two-person neuroscience, how two brains combine together through behavior, and to test long-term trajectories of human development, what enables some children to thrive amidst hardship. So this is my lab, my students who conducted these studies, our funders, and thank you for listening.